welcome to all of you. We are looking forward to now get an exposure about the dynamic topic zebrafish as a model organism for CNS drug discoveries. As the zebrafish Danube radio is increasingly utilized as a powerful animal model in neuropharmacological research and in vivo drug screening. This makes zebrafish a very useful tool for drug discovery and identification of discovered molecules. So this session is definitely going to be very innovative and informative.
So one of the important aspects of CNS drug discovery is the behavior analysis. Can this be modeled in zebrafish model? The answer is yes. So if you look at the timeline, how much zebrafish has contributed, hallucinogens and drug withdrawal testing are actually kind of established in zebrafish model. Where we have contributed from our lab is developing a model of epilepsy-induced cognitive dysfunction. And we are actually developing further with the chronic model as well, which we have published very recently. The good news is zebrafish allow high throughput screening. So the recent trend is COVID-19 research, and you will find studies where people actually using zebrafish model to identify and validate antiviral drugs for COVID-19. So this is in a nutshell how they did to look into the effects. So that was about in detail why, what, and how zebrafish. I'm giving example of epilepsy, and as many of you know, it's a chronic disorder, which is basically a short circuiting of the brain, you can call it, and which leads to muscle jerks and severe to prolonged convulsion. And in a nutshell, you can call it as an electrical storm in the brain. And these are some symptoms. I'm not going to go detail, but you might have seen many people who has different symptoms when they are suffering from epilepsy. The very common one which we observe is uncontrollable jerking movements in epileptic patient, and that's really alarming. I know there are a lot of stigma and misconception around this disease as well, but we probably talk some other time in detail about it. So I'm trying to tell you guys like how this disease condition can be modeled in zebrafish. So when you develop any animal model for drug discovery, you look into whether what is the molecular mechanisms involved in, what are the phenotypes of it. When I'm talking about molecular mechanism, I'm talking here where we know, epilepsy or anti-epileptic drugs, they work through GABA, sodium channel, potassium channel, calcium channel. There are new drugs like perampanil, which is AMPA receptor inhibitor. And then you have electroencephalographic EEG, which is a gold standard for diagnosing and looking at somebody has epilepsy or not. And then looking at the phenotype, very typical behavior related to disease. So what we try to understand, okay, these are the key pathological markers of the disease, and this needs to be modeled in any organism which can closely mimic the disease condition. So what we need for an ideal model is use a model that recapitulate some of or all of the observed pathologies. So we do not have any single model or animal model which is extremely perfect, which is not 100% similar, but it has closeness to it, like I talk about like 70 or 80%, which is close enough for disease modeling, and which can answer both genetic as well as phenotypic basis of the disease. I think this is again very, very important, and it allows system to test the potential treatment which could be lead for drug discovery in that specific condition. And when we talk about modeling the zebrafish in epilepsy condition, I said, okay, pharmacologically, we can test drugs using this system. We get a behavior parameter, which is phenotype I was talking about. Can we do electrophysiology in that? Can we do the mutants, injecting the morpholinos in that? And does it allow doing gene editing in zebrafish models? And the answer is yes, electrophysiology can be done in zebrafish. There are a number of papers which talk about the multi-channel EEG recording in adult zebrafish models and also in larvae. And this is another system which talk about printed chip which are actually placed on zebrafish skull or the head, and then the EEG can be recorded by testing different drugs. So that's a good news that you can actually study electrophysiology there. When you look at the NIH, the National Institute of Health Anticonvulsant Screening Program, I'm trying to highlight it uses maximum electroshock and pentylene tetrazole model, and also 6 Hertz model, and so on and so forth. We can use the same pentylene tetrazole test in zebrafish as well. Having said that, NIH hasn't given a green light for including zebrafish model in this anticonvulsant screening model yet, but still I will tell you how zebrafish actually got approved directly into the clinical trial approved by FDA. And these are the typical example of two different types of models in zebrafish. One is chemical induced and one is genetic induced, and I'll talk based on my results further about it. So this was the one of the important paper which talk about morpholino versus mutant, and they say the reverse genetic injury screening reveals poor correlation between morpholino induced and mutant phenotypes. So they say mutant phenotypes are much more better genetic organism for epilepsy study in zebrafish compared to the morpholino. So that's one. This is one of our study, and I'll be talking more about this, which is developing the zebrafish model for epilepsy-induced cognitive dysfunction. We have taken this pharmacological, biochemical, and behavioral approach. Then what we have used is pentylene tetrazole model. We have injected intraperitoneal in zebrafish after anesthetizing it. Then we have recorded the behavior of the zebrafish in a tank, and this is how the behavior analysis looks like. And we have used the PenLab smart software for the analysis. We looked at seizure score, onset latency, as well as the locomotion parameter in that. And this is a typical video. I hope I can show you guys. So this is how the typical zebrafish seizure behavior looks like when you inject 220 milligram of zebrafish. These are the different seizure scores you will be able to see in them. So when you start, it starts with the short swimming at the bottom of the tank, because fishes are a bit anxious after injecting pentylene tetrazole. Then after that, it will start or increase the speed, swimming speed in the zebrafish. That's typically abnormal behavior. If you keep a normal fish next to it, you can clearly see the difference. You can still see it increases swimming activity. And then it moved to the seizure score 3, which is burst swimming, left and right erratic movement, which is again not normal in normal healthy zebrafishes. So it shows a typical behavior what you see in the people when they have their epileptic seizure attacks. And finally, you also see the circular movement. This is very typical of the zebrafish epilepsy behavior in which we mark as the score 4. And then further to that, you can also see a seizure score called seizure score 5, which is a typical clonic seizure-like behavior, which comes with complete body rhythmic muscle contraction. And that really, crazy behavior you can see in zebrafish. So how we recently published another book chapter which actually talk about these behaviors, and these are the behavior patterns. I'm again not going to go in behavior because you already seen that in the video. And then this is how you develop the seizure behavior patterns. So this is a control zebrafish behavior when we haven't injected pentylene tetrazole or proconvulsion in that. And this is where you inject pentylene tetrazole, you see the abnormal or abrupt behavior in them. This is tracking from the smart software. So these are some of the results. If you see standard drugs like phenytoin, oxcarbazepine, gabapentin, diazepam, and rivastigmine, they able to control the seizure scores compared to the negative control, which is only PTZ. And you can also make it from the behavior. This is PTZ behavior. and they are coming to normal in the drug-treated animals. And these are the behavior we see to check their learning and memory. If you look at 0 day, 0 hours, it took about 32 seconds for these zebrafishes to reach the deeper chamber, which is part of the learning and memory behavior. Fishes normally prefer the deeper and the bigger chambers compared to the small, long and the right arm. So it takes about 32 seconds for this zebrafish to reach in this chamber. Whereas when we tested the retention of memory after 3 hours, we could see these fishes actually move faster at 16 seconds, and they quickly enter to this chamber and stays there. And then when we check it after 24 hours, you can see these zebrafishes actually move quickly because they remember there is a deeper chamber on the right hand side, and they don't take any extra time and then quickly move there. So this is a typical behavior we see from zebrafish, and it confirms their learning and memory behavior. So yeah, this is done by my PhD student, Brandon. All right, and this is how the typical patterns looks like. You can see the zigzag movement. They spend more time here. They're trying to explore. Sometimes they also go to this left arm, or we call it wrong arm, and then finally they reach. Once they reach there, they try to stay there. But when you look at the 24 hours, the time taken was very less. So this is, I'm not going to go in detail. If anyone knows more information, you can contact me later. But we calculate inflection ratio, and this is where we draw. The important thing, if you look at 3 hours and 24 hours, the improvement in the learning and memory behavior compared to the negative control, and can also be very much seen from the tracking behavior. And then we also looked at the chronic model of the epilepsy, and we have used pentylene tetrazole subconvulsive dose for repeated administration up to 10 days, and the single dose of canic acid, which is used for chronic epilepsy in rodents. But surprisingly, we found canic acid could not sustain the epileptic behavior in animals until 10 days, and the zebrafish recover, and they do not produce a chronic seizures in there. And this is again another video which shows how zebrafish learn to navigate and move to the different compartments. This is a very, I will say, stringent or more complicated learning and memory behavior, which basically work on working memory in zebrafish, and they reach the target in 18 seconds. These are the trained one. Normally we allow training for 10 days before they go. And this is how it looks like, a vehicle, probably they learn, and then those who have impaired memory, they take longer time. And again, canic acid also took longer time. So yeah, so PTZ screening, which is for high throughput screening. You can use 96 well plates. You can put in 1 larvae in each well, and then expose to 20 millimolar of pentylene tetrazole. And then you can observe using the camera or specific systems. I'll talk about it in a minute. And the another model we have used is the knockout model. This is in collaboration with Montreal in Canada. And we have used 3 knockouts: GABRA1, GAMMA2, and SCN1, which are basically genetic epilepsy models. We have used this Genu Vision knowledge information technology system for observation, and our knockouts, we have recorded them that on the light, and then observed the behavior for 2 hours, and then we have recorded the maximum acceleration of these animals. So in this drug discovery, I think what we have used about 68 synthesized derivatives, and we use PTZ model as a primary screening model. We got about 11 hits from there, and then we have used these genetic models to further look at where, and then we got 2 hits here in GABRA1, 4 hits in GABRA2, and 7 hits, which we are doing for the analysis. So yeah, this is in a nutshell what we have done. We have done behavior analysis. We have used mutant models, and then we also use calcium imaging to check whether the effect is because of the epilepsy or it is just some brain activity. So just for that confirmation, we have calcium imaging as well. And these are some of the results which are still unpublished, under consider review. you can see the wild type homozygous, which are mutants, and actually has increase in acceleration, and where the treated animals actually control these acceleration in animal models. So this, in a nutshell, actually leading to the personalized care. When I say personalized care, if a patient is suffering from genetic epilepsy, the genetic mutant can be found out, then because a lot of these patients do not respond to a lot of known anti-epileptic drugs, they are drug resistant, and for the same reason, zebrafish model actually play a very important role to check the effectiveness of anti-epileptic drugs in zebrafish model. And once you confirm a specific anti-epileptic drug is working against the genetic mutation in zebrafish model, then it can be given back to the patient, and a lot of actually personalized medication care or epilepsy care is done by that method. So zebrafish finally earned their stripes for the drug discovery, and this was when the first drug which actually bypassed the preclinical testing on rodents and made it to the phase 1 trial of the clinical trial. And this is coming from the Baraban's lab in UCF in US, where they have used this drug chlamysol. Basically, it's a drug repurposing, and they have found very interesting results in Dravet syndrome, and now it is in clinical trial. So this is the paper, if anyone interested, which published in Brain by a Scotch group, and they have shown the chlamysol and the modulators of serotonin signaling actually suppress seizures in the Dravet syndrome. Dravet is a child epilepsy disease where children cannot survive beyond the age of 10 or 12, and this drug actually kind of hope for those kind of patients. So finally, zebrafish are actually helping to fight epilepsy in both genetic and non-genetic models of epilepsy, which can be utilized in the patients. That's all from my side. Thank you for that, and I thank my funders, collaborators, and my lab for these results, and I think we are actually moving forward in this direction. Thank you everyone for your attention. That's all from my side, and I'm happy to take any questions.
Very well explained, sir. Thank you so much. We'll also try to remember all these words: refine, replace, and reduce in our research. And also, we'll try to incorporate the zebrafish model in our institute. And now we have some questions from our participants. May I ask, sir?
Yeah, sure.
Okay, so the first question is: use of zebrafish as a model organism began in 1960s. So why there is still a delay in modern research in the field of anti-cancer drug or anti-diabetic drugs?
I think this question is more related to the target identification. A lot of disease, I'll just give an example of, Alzheimer's disease, or you talk about diabetes and others. I think I personally feel there is more of an individuality factor rather than a disease itself. The condition or the mechanism in each and every patient is actually different. Just take an example of Alzheimer's disease. Although we know there are a lot of hypotheses like beta amyloid, tau hypothesis, free radical generation, or neuroinflammation, but all these conditions actually not found in a single patient. So there are one patient has predominantly beta amyloid in them, whereas you find another patient which has more of the free radicals on oxidative stress. So the makeup of individual patients of that disease they are actually showing, I feel is not really same, and this is the reason why we have these problems. So I think the problem is more with the target identification. Once you have the target identified, and then you can mimic that target in zebrafish model, the drug discovery will be at faster pace. So I will not say it's a problem with the animal model as such, but it's about the target.
Okay, thank you, sir. I think we can also incorporate computer-aided drug design in this. And
yeah, definitely. If you look at the industry, the way they work, now they have reduced all the cell-based or the animal-based assay in their initial screening, but they are more into simulation and computer-aided drug screening before they go to the hits, and then they check further in the animal models. So I think definitely that is the way industry is following. Maybe it's a time for institution also to think about it and start somewhere there.
Okay, sir. So my next question is relevant to this, sir. In small setup like our institution, how we can incorporate this zebrafish model?
I think the zebrafish model is good to go, especially when sometimes we have problem with the animal facility with rodents and all. For zebrafish, I think you need not to worry. With a single tank in your own lab or in your office or in your home, you can kickstart your zebrafish research. And I think Sanjay is here. I'm more than happy if you guys need any help and support from my end to help you to set up and kickstart your work on zebrafish models.
Okay, thank you so much, sir. And now I request my participants, if they have any queries regarding this topic, they can raise their hand or they can unmute themselves and ask the questions.
Okay, anyone got any question? Yeah.
Hello.
Go ahead.
Dr. Frooq. Just curious, how do you administer the drug into the zebrafish?
Hey, hi, Mickey. Good to see you here. We use both the methods, but I think being a pharmacologist, I'm more interested to inject a known quantity. And for the same reason, we have optimized our intraperitoneal injection using microneedles in zebrafish. So that's something we use. But having said that, we also use by directly dissolving the drug into the tank, and that give you molar concentration rather than mg per kg concentration for each and every animal. I hope that I answer your question, Mickey.
Yes. So is there any difference between, like, if you inject the drug into the tank, that would be oral route of administration, and if you inject directly into the peritoneal fluid or the blood, would there be any difference in the
Yeah, I think if you are using for screening point of view, I will not be so worried, because the endpoint in the screening is whether it is working or not. That's your legitimate question. Yep. But when you are looking at specific, whether what quantity, or if you're looking at dose dependent activity, I prefer to know what quantity actually going into the body. When you dissolve into the water, you do not know how much is consumed by individual zebrafishes. And another alternative for that, I think a lot of people, we don't really do that, but some researchers have developed oral gavage administration of whatever your test compound in zebrafish as well. But my personal preference is intraperitoneal or dissolving in water. So as I said, dissolving in water is good for screening, but if you want to know the dose dependent activity, I think intraperitoneal injection is the way.
I see. It makes sense. That's very interesting. Actually, I'm interested in exploring this zebrafish model for airway diseases, which is my research interest.
Great. Yeah. I mean, we can talk more if you want.
Yeah, sure. Thank you.
Most welcome.
Thank you, sir. Thank you, ma'am. If any other participant have any questions, they can ask. Okay, I think there are...
excuse me, ma'am. Can I ask a question to sir?
Yes, ma'am. Sure.
Good afternoon, sir. Sir, can we use this zebrafish model for transdermal drug delivery?
The answer is yes, and I think there are a number of papers where people have used that. Transdermal. Wait. I thought just nano formulation.
Yes,
I think that could be challenging, but I don't think should be a problem. But you need to have a patch which is very tiny. That's a challenge. Okay. And that too, I think nowadays using 3D printing, you can actually do that, but the quantity will be very small. You can talk to Dr. Tahir further about the 3D printing of the some of the patches, and then at the same time it should be good enough or detached. It should be detached to the skin of the zebrafish. I think that's again very, very important. Probably micro needles will be a good option, because that will go into the screen and not on the superficial, because you need to consider the loss of drug if it is just very superficial. And I think attachment to the skin of the zebrafish is again very important. But I think if you personally ask me, rodents or mammal models like guinea pig or rabbits are much more preferable when you're talking about skin diseases, because with
Yes, sir.
one model you want to know the ultimate answer rather than trying on various models. Yeah.
Okay, sir. Okay, sir. Thank you very much, sir. So there is a query about that the drug dose. When we give drug dose in the tank model, or when we are applying it intraperitoneally, that it will be calculated. But does it is not like कि अगर if we give in the tank mode, so we have to give a lot more amount of drug in that part.
Yeah, that's correct. But so what I prefer, when you know the compound is a pure compound, is good to go intraperitoneal. So there's no worry about that. Because if you're using extracts, I will prefer to dissolve in the tank. And I think in India, a lot of institution, they use extract for screening for various projects. I think
Yes, sir.
dissolving in water is the good way. And you can have just 1 liter tank, and accordingly you calculate your drug. You don't need a very big 10 to 15 liter of tank, and that time your extract will be like finish in one side. So you can use one tank, and you can actually reuse the same tank for exposing different zebra fishes. But there is a matter of how much time you will be exposing them into the tank to get the drug, the maximum absorption from the drug, and all those are the question. But if you're planning a specific project or a specific
Yes, sir.
experiment,
Yes,
I'm
sir.
more than happy to support or help in that.
Okay, sir. Thank you very much, sir. I just want that because we literally in our preliminary stage, we just use extracts over here. Na? Thank you very much, sir, for
Yeah. No worries.
clearing my queries.
Yeah, no worries. I think there is a question in the chat which talk about from Alishan. Sir, reverse genetics also works in zebra. So I'm not a genetist, so I cannot answer that question. If genetics works, probably reverse genetics also works. So that's my answer. But don't quote me anywhere because I'm not an expert on that.
Sure, sir. Thank you, ma'am. Thank you, sir, for this answer, for this very informative and very interactive session. We have one more question, sir. Sir, what are the alternative economic models, or we can say economic methods we can use?
Alternatives are using worms. That's another alternative where you can. But I think, having said that, and also in my presentation, I think these models are much more suitable for a lot of CNS related diseases. And fruit fly is again. But again, if you look at fruit fly compared to zebrafish or worms, are more complicated because they have wings, they can fly. So you need a control system actually to manage them. Each time you want to administer any drug, you need to anesthetize all of them using some anesthesia and all those things. So I will say worms and Drosophila are two other models which are actually getting popularity in the CNS drug discovery or overall drug discovery or animal modeling as such.
Okay, thank you, sir. So here is one more question: that what kind of light do you use for zebrafish?
So it's a normal white light. You can optimize the lux because if you see the zebrafish basically originate from the Ganges rivers in Himalaya. That's the main origin of zebrafish, and now it went to all over the world, including US and other places. So it's a very much local. You need a natural light, and what light we prefer the same light, and everything fine. But I think the light and dark cycle is very, very important. If you do not maintain 12 to 12 light and dark, or 10 or 14 light and dark, then zebrafish may get stressed, or they may have a sleep related problem, and then you may see the problem in your behavioral studies, specifically if you are using behavioral studies, that you can. So a light cycle of 12 to 12 normal light is absolutely fine, which works for zebrafish.
Okay, thank you so much, sir. Thank you. If any
Excuse me,
any more questions. Okay.
excuse
Yes,
me,
sir.
Indu.
Yes, sir.
Thank you so much, Dr. Farooq, for your informative session. And definitely, I will discuss regarding this establish of this Zebrafish model in our institute. And I hope you will give your best for us.
Surely, most welcome. I think now Dr. Sanjay has said, though, you will get a Zebrafish system in your lab at least.
Yes, sir.
Okay, okay, definitely.
Thank you, Sanjay sir, and thank you so much, Farooq sir. We truly appreciate that you explained this topic very well, and every minute of your lecture was very informative and very interactive. So we will try our best to incorporate this model in our institute, and we are very grateful to you for sharing your experience and your knowledge. Thank you
Thank you
so
so
much,
much.
sir.
It was a pleasure talking to you guys. Thank you.
Thank you, sir.